Welcome. Today, we're diving into the quality assessment of sustained and controlled release formulations. This topic is crucial for ensuring medications work effectively over time. Dr. S.K. Singh will guide us through the key concepts and methods behind evaluating these advanced drug delivery systems. Let's get started. Let's start by understanding what sustained and controlled release formulations really mean. Sustained release is all about releasing the drug slowly over time, so patients don't have to take it as often. Controlled release takes it further. It delivers the drug at a precise, predictable rate throughout the dosing period. The goal here is clear: improve patient compliance, keep drug levels steady in the body, reduce side effects, and ultimately make treatments more effective. When we talk about global regulatory expectations, we're looking at a complex but essential framework. In the U.S., the FDA provides guidance on modified release products, focusing on how we develop dissolution methods and follow product-specific rules. Over in Europe, the EMA offers its own guidelines for modified release dosage forms. Then there are the ICH guidelines, which cover everything from pharmaceutical development and quality risk management to analytical procedure development and validation. Finally, the USB gives us specific references on dissolution and drug release to ensure consistency worldwide. When we talk about critical quality attributes, or CQAs, we're looking at the key features that define a drug's quality and performance. These break down into several categories. Functional attributes cover how the drug dissolves, how it releases over time, and the risk of dose dumping. Stability attributes focus on how consistent that release remains, along with sensitivity to moisture and the integrity of polymers involved. Physical attributes include things like appearance, weight, hardness, and size. Finally, chemical attributes ensure the right amount of active ingredient is present, uniformly distributed, and free from impurities. Now, let's focus on assay and content uniformity. These two concepts are critical when we're measuring the amount of active ingredient in a product. Assay tells us how much of that ingredient is actually present, while content uniformity ensures that this amount is consistent across all units. Together, they guarantee that every dose delivers the intended effect, no surprises, no variations. This is essential for both safety and effectiveness in any pharmaceutical or chemical product. Now, let's focus on physical quality assessment. This is where we evaluate the tangible aspects, the materials, the build, the feel. It's about making sure what we create not only looks good, but also stands up to real-world use. Physical quality is what turns a good idea into something reliable and lasting. We'll explore how to measure and improve this critical dimension next. Let's talk about why dissolution testing matters so much in regulation. Dissolution often stands in for how a drug performs inside the body. Different USB apparatuses help us test various dosage forms. For example, USB-I for capsules, USB-II for tablets, and others for more complex or extended-release products. The main goals here are to evaluate the release rate and mechanism, ensure batch consistency, and predict bioavailability. This testing is a cornerstone in meeting regulatory expectations and ensuring safe, effective medicines. Let's walk through an example of how dissolution is calculated. This shows us how a substance breaks down or dissolves over time under specific conditions. By working through a real calculation, we get a clearer picture of the process and why it matters. It's one thing to understand the theory, but seeing the numbers in action really brings it to life. This example will help us connect the dots before moving on. Let's talk about drug release kinetics, how a drug leaves its delivery system and enters the body. This process shapes how quickly and how long a medication works. Understanding these patterns helps us design treatments that hit the right balance between effectiveness and safety. Whether it's a slow, steady release or a quick burst, the kinetics determine the drug's impact over time. It's a crucial piece of the puzzle in developing better therapies. Here, we see a simple table showing how much drug is released over time. The amounts increase steadily: 40 milligrams at 2 hours, 80 at 4, and so on, up to 160 milligrams at 8 hours. This pattern illustrates what's called zero-order release. That means the drug is released at a constant rate, no matter how much is left. It's a steady, predictable process, exactly what we want for consistent dosing. Now, let's talk about the similarity factor, or F2. This metric helps us compare two sets of data, usually test results against a reference. It gives us a number that tells how closely they match. The higher the F2 value, the more similar the profiles are. It's a simple, effective way to measure consistency and ensure our results align with expectations. Understanding F2 helps us make confident decisions based on reliable comparisons. Here, we see dissolution data laid out over different time points, from 2 hours up to 12. The table compares reference and test results side by side, showing how closely they match at each interval. This kind of data is crucial for calculating the similarity factor, which tells us how alike the two samples are in their dissolution profiles. It's a key step in ensuring consistency and reliability in our testing process. Let's talk about dose dumping, a serious issue where a drug meant to release slowly instead floods the system all at once. This can happen for a few reasons: alcohol interacting with the drug, failure of the polymers that control release, manufacturing defects, or damage to the coating. For example, a formulation expected to release 20% in 2 hours might suddenly release 65%. This spike raises regulatory red flags because it risks toxicity from too much drug in the bloodstream too quickly. When we evaluate a product's stability, we look at several key parameters. These include the assay, dissolution, impurities, moisture content, and physical integrity. Our goal is to ensure there's no significant change in the release profile throughout the shelf life. We test under different conditions: long-term, intermediate, and accelerated, following established guidelines. This thorough approach helps us understand how the product behaves over time, ensuring safety and effectiveness from start to finish. To wrap up, dissolution testing stands as the most crucial quality check for sustained and controlled release products. Understanding drug release kinetics lets us uncover how the drug behaves and performs. The similarity factor, or F2, is vital to prove that different profiles match up. Assessing dose dumping is a must for safety and regulation. Plus, all analytical methods need to be validated and woven into a strong quality system. Altogether, this thorough approach guarantees consistent therapy, patient safety, and smooth regulatory success. Thanks for joining me today. If you need any more support or have questions, please don't hesitate to reach out. I appreciate your time and attention, and I look forward to continuing the conversation. Thank you.